Hello, everyone, and welcome to our event. Today we'll be talking about XRD and XRM, but before we get started, I just wanted to give you a quick little tour of our venue. So if you have joined us before, welcome back. If not, then welcome for the first time. This event platform is kind of nice because it is very similar to a real-life platform, or a venue. So on your side of your screen, you'll see some vertical icons. That's how you can navigate around the platform itself. There is a reception area. That's where you likely started. And then to get here, you had clicked on stage. So within the stage, that's where we're going to give this main presentation. Now on the other side of your screen, you're going to see a few different chat boxes or functions. One is just a general event chat where you can type any messages you'd like that everybody can see, or if you have a general question. But there's also a Q&A tab. So if you click on stage, then you can type your questions in there. So that's the preferred way of submitting your questions. Go ahead, type those questions in, and then we'll check them out and get them posted up. So at the end of today's presentations, and we'll have two of them, that's when we're going to go through the question and answers. So with that, I'm going to go back. We're going to finish setting a few things up, and we'll return in just a moment. All right. So welcome to our event of how XRD and XRM can be used for optimizing the dosage form and drug delivery and pharmaceuticals and formulations. That is a long one, and I'm sure that I totally screwed that one up. But to just introduce our speakers today, we're going to bring them on stage now. So the first one is from RJL. Hello, Marcus.
Hi, John.
So Marcus Henneke, he is our expert in X-ray microscopy and the use in pharmaceuticals. So he'll be giving our first presentation. And then Nate Henderson. Hi, Nate.
Hi, everyone.
Nate is our expert in X-ray diffraction and its use in the pharmaceutical and general, you could say, health industry. So welcome and thanks for joining us today.
It's good to be here.
Yes, it's good to be here.
So again, if anybody has any questions for Marcus, Nate, then go ahead, submit those on the side of your screen. Please use the Q&A, which is found on the stage area, and we will get to those at the end of the presentations. So to kick things off, we're going to hear from Marcus.
So, John, I think you can see my first slide. Is that right? Obviously, yes. So I'm going to present about X-ray microscopy, XRM, which is, in my opinion, a powerful tool for the analysis of pharmaceuticals and formulations. I'm going to show you some case studies and success stories. And first, I'm going to introduce myself. I'm Marcus Henneke from the company RGL Micro and Analytic. Just RGL is enough. And our vision is to bring instruments and analytical services to the market from a single source. From the brands and instruments we are using, I'm going to speak today mostly about the X-ray microscopy systems brought by Bruker Microcity, but we also use for customer contact scanning, tabletop semi-leaks and particle analysis instrument, and even specimen radiography. Our laboratory is equipped with a accreditation according to ISO 17025. Okay, so that's enough for the introduction of myself and my company. I'm going to jump and give you a few fundamentals of XRM, so we are on the same track and have a basic understanding of the method. So X-ray microscopy, or sometimes called microscopy or microtomography, it's basically the same. It's a three-step process. And the first one is the data acquisition, and the second is the reconstruction. And then finally, that's where you want to go, that's the data analysis. And I'm going to present you with those steps so you have an idea how to use the instrument. So first one is data acquisition, where we use an X-ray source, which is here on the left-hand side. And the X-ray source is pointed towards a detector, which is that yellow in here. And in between, we have our sample. And this sample is rotating actually around the z-axis. And the screen is just observing the projection image of the sample into the detector. And I've brought you here one example. This is a German candy, a Haribo strawberry candy. Likely there is something similar in the US. Many kids like it here. And you can already see the projection images. There is something inside. And we will see what is going on here in the next slide. One thing I like to point out is the magnification. It's very simple in the system. Just by moving the sample towards the detector, towards the source or detector, we can change magnification. For example, if we want to have a large field of view, we go close to the detector. And if you want a high resolution, we will move the whole thing to the source, which leads to a small field of view, but a high magnification. And then we acquire the projection series and do the reconstruction. This is normally done by a Feldkamp back projection algorithm. And the result is pretty much nothing than an image stack. So we have an image stack which represents the 3D volume of our sample, and it is coded in isotropic pixel size. Here's a small animation shown. This is our sample in vertical cut, and you can see here the horizontal cuts, and the red line indicates where we are actually in the sample, and this corresponds to the cross-section shown on top. And we see here there is a large bubble inside that sample. It's probably no harm for anybody, but this is only candy. And then we jump to the step number 3, which is data analysis. And this typically starts with virtual cross-section images or 3D rendering, which is then shown here, the 3D rendering. And this can be pretty useful, for example, for failure analysis of medical devices and applicators. You can analyze density distributions inside, or inclusion and particle analysis can be done. And you can do dimensional measurement, so-called metrology. That is useful if you want to compare, for example, your medical device against the cat, the cat drawing, and see the differences. Okay, that is the fundamentals. I'm just jumping to the instruments that we propose for pharmaceutical industries, and there is a very nice instrument lineup from Bruker Microcity. They offer desktop instruments in the SkyScan line. They are quite compact in footprint and easy to use. And the 3 main instruments that I would recommend are the 1272, which is a very high resolution desktop machine, or the 1275, which is a fully automated scanner, which makes it really easy to use for beginners. Or if you are more in medical devices and need a large sample size, you can go to the SkyScan 1273, which offers samples up to 25 centimeters. But this we can talk later if you are interested. I'm going to go in medias res and show you some nice examples, and we start with solid formulations. So first thing we look at will be, how does the reconstructed volume of a tablet scan look like? And it's typically shown in orthogonal cuts. If you have the software, you can browse in any layer here and look at the details. But what I want to point out is a detail here on the right-hand side, which is a magnification of that part. And you can see a very important lesson, that the grayscale in this image is equal to the density, to the local density. You can observe this easy by looking at the air around the tablet. This is coated almost in black. So this is a low density. And here we can see a coating of the tablet. It's a really thick coating. And this is kind of a medium grayscale, which is a medium density. And inside the tablet, there are mixed densities, but you can see, for example, those high bright particles, and they are high density particles. So remember, when we look at microcity images, micro X-ray microscopy images, grayscale is equal to density. And we can do a few things with that. XRM is, for example, used assist formulation development. That would be a typical R&D topic. Then we had a number of projects where we control the quality during scale-up. It's very important when you implement from development into production. And later, you can look at batches. For example, you can look at the inter or at the intra-batch variability from your product. And what we look at is typically the homogeneity of tablet, density distribution. We can look at cracks or other structural defects. And then the more quantitative things, we can do porosity analysis. We can easily assess the thickness and therefore the quality of the coating. And we can do volumetric phase analysis in a quantitative way. And that's where we want to start because it teaches us a basic lesson of the image analysis task that we have to do. So let's take this tablet example and look at one cross-section, only at one cross-section. So what you know from image analysis, there are many grayscales inside, and there's a representation called grayscale histogram. This is actually shown here. So this goes from black on the left side to white on the right side. And all the grayscales that we count in that image are actually represented with their frequency. So in order to select a phase, we need to do a segmentation with a threshold. In this case, I have chosen the bright phase. So I'm going to use a threshold which is close to white on this side. And once we apply the threshold, the image will be divided into structure, which is white, all the object is structure, and black, which is just separated space between the structure. And the good thing is with this minus image, we can let our computer run and compute volumes and sizes and thicknesses and also on. So let's just use this example and see the result for this green phase, which is, by the way, in this tablet, the API phase. So we can do a volume and structure analysis of the API phase, and we can even visualize that. So this on the left-hand side is a 3D rendering with the visualization of the API phase. On the outside, I have visualized the coating, and the rest of the matrix have been set to transparent. So we can then calculate the total API phase. In this case, is 10.9 volume percent, and the mean particle size of the API is 0.16 millimeters. But we can drive that even further. We can look at the API size, API particle size distribution. And then we realize there is a large chunk is around 0.1. And then we have some large particles which even go up to 0.5 millimeter in size. So this is pretty important information, and it can help you optimize your powder preparation before tableting. Now, you may ask how this is precisely computed, and I have a very simple explanation for you. We use a method called the local diameter method. And this is easy to explain with a human person. It's a two-step approach. First step is we compute the medial axis by a step called skeletonization. And you see, this is the medial axis. It can be considered to be like the center of mass at any given point within the structure. And once this axis is established, we can start computing local diameters by blowing up spheres with the middle point on the medial axis, and therefore we can compute at any point within the structure the local diameter. This is a large one close to the belly, and a smaller one here at the arm or at the foot. So this approach is very strong. It is extremely useful also for the coating. So let's use the same tablet again and look at the result of the coating. It turns out that the variance of thickness is the most important parameter apart from the thickness itself, the variance, and I explain you why. So we have the coating here in red, and we have calculated the total coating volume, which is an important parameter, and the mean thickness. But actually, the mean thickness doesn't tell you anything whether the coating is missing at some point or too large at another point. So we look at the spread in the coating thickness histogram and look at the variance, which is which is this part here, the width of the peak. And the wider the peak, the more, the less is the quality of the coating. So you want a nice and narrow peak here, and you can easily compare different coating methods by scanning the tablet and looking at those systems. And tablet coating can be even driven further, because it's still an overall view on the coating. We can look locally if there's a problem with the coating. We can look locally by using a color-coded thickness map in a 3D in the 3D image, because this will reveal the weak points of the coating. What we see here is a tablet, and we have a part of the coating we have cut out, so you understand what is going on here. And then we have color-coded the coating to be blue in the area where it's thin, and then green in the thicker area, and even red towards the most thickest area. And you see the strength of this concept. We'll show you immediately if you have any weak points here. And you can, for example, from this, you will understand that the imprints in a tablet are always critical points on the edges. And you can learn this very easily applying this method. So moving away from coatings, we know that cracks in tablets are a common problem. And I've brought to you one example with cracks. You can see the cracks are in this image displayed in red, even here on the rendering on the right-hand side in red. So why is this happening? In this case, pressure was obviously too high. So we apply the tableting pressure in this direction, and then the ingredients of the tablet start to form layers, and those layers are actually orthogonal to the pressuring, to the force direction. And then those layers start to float on each other, and they will form cracks after the stress is released. And this is a typical picture of a tablet that was tabletized with a pressure too high. And we can use actually now quantitative assessment to steer our production. We will do, for example, tablets with different tableting pressure. And we will just calculate the red volume, the air volume actually inside the tablet. This is corresponding to the crack volume. And we will draw the volume over the tableting pressure. And we find out typically that it will reach some kind of minimum. This is the best, this is the optimum and best tableting pressure for this formulation. So it's quite useful. XRM can help you to find the optimum pressure on tableting machines. Another thing that is regularly done with XRM is to compare drugs which are the same medicine, for example, original and generic drugs. So I've brought to you one example from a tablet. It's the same medicine, it's just different manufacturers, and this one is the originator. And you can see there are lighter grains and there is a darker matrix, so we don't know exactly what it is, but we know at least two faces are visible and a coating in micro-CT. And now let's have a look at the same medicine from another maker. It's a generic drug. And you can see this one looks totally different. So we have a smooth matrix and there are no particles to be found. And the reason is very simple. Although I have not talked to this manufacturer, it's very likely that this one just mixes up the powders, and this one probably goes for a micronizing method. They mill actually the powders to a very fine grain structure and then start tableting. It will work as well, but it's of course a little bit different structure. And in this case, the kinetic of the release is not the issue. So probably it works both in a similar good way. But if the release kinetic is important, this will behave different than that one. And then we have a generic product 2, and this guy cooked the recipe of the originator quite well. Very similar structure. And then we have another company. Wow, that's an interesting one. Here you can see the crack example again. Those lines which form the thin lines which form from top to bottom here, they are all cracks. So we can think that the tableting pressure in this scenario has been set too high. But you can even see further things in this formulation. If you look, there is this dark air structure, but there are other dark structures inside. They are not cracks. They are more like particles, longish particles. I show you with my marker these particles. There's another low density phase suddenly, and this points to me that this is a fat or grease or phase which the manufacturer put inside the recipe because he realized that the taps are cracking. So he put something inside to make it glue again. So that is this guy should have used definitely microcity to make it better. And then we have a generic drug number 4, also similar, but you can see already this guy didn't apply coding, which tells me the release of the drug is not so much a problem. Another topic that has been worked a lot in the last years is the lyophilization of drugs. So lyo drugs are basically freeze-dried. They come in powders and typically in a glass bottle or even in the airtight blister. And on the left-hand side, I've picked you an image with a very large-scale freeze dryer. So what they do, they put in the vials here with the liquid API, and then they start freeze drying it to make it durable. And at the end, they have to make it airtight in packaging by using those sealings or in a blister. So typically, this drug is then dissolved with, for example, water for injection before use, and it can be oral forms or more likely even parenteral donation forms. And in the past years, we have developed a method to scan the lyophilization cake through the glass. And this is not immediately understandable for you, but this is a difficult task because the glass is actually made of silicon and oxygen, and the lyophilization plate is only some carbon, so very low dense phase. So most of the X-rays get absorbed in the glass, and only a few of the X-ray flux is left to image and make the contrast for the actual object. So we have worked a lot on that to make it better, and came finally up with a beautiful solution to make actually scans of the lyophilization plate within the glass. And you can see here, this is really a beautiful structure. And the scan is really crisp and the contrast is good. But also in this case, the lyophilization cake is pretty good. Now we can use this not only for viewing, we can use this to study the influence of the process parameter on the cake structure. So we use lyophilization cakes from the procedure. They can be from different process parameters or just from different regions within the freeze dryer. And then we compute the density map in cross-section in 2D. And typically, if in the lyophilization process is not good, we will have a very inhomogeneous structure, especially from top to bottom. It will vary a lot. In this case, it's a good example. And then we can study, dependent on the height position, we can study the pore size. It's quite good. It's varying between 55 and 65, but it can vary from something like 20 to 150 or so. That's then very inhomogeneous. And even porosity in this example is very well established over the whole height. So it's a good example, and XRM can help you to find the optimum process parameters for the lyophilization instrument. And as we speak about those glass vials, there's another thing that X-ray microscopy can help you. I'm pretty sure you all know that foreign particles are a major concern in all pharmaceutical products, and it's very difficult actually to assess a foreign particle that is buried somewhere inside the lyo cake. But we can use now this method to actually do some ultra-fast image acquisition using a continuous rotation in order to detect the foreign particles in the finished enclosed product. And we had recently a study with the university and the manufacturer here in Germany to bring that really down to quick scans in order to be close at line and having a significant number of samples. And you can see this is a 60-second scan, so pretty fast. The lyo cake is almost homogeneous, but you can see there are some foreign particles that are embedded here. And even with the 20-second scan, it's still very nice in contrast. It's going down then into 10 seconds. Still, the largest one are possible to detect within the glass vial and without opening. So that is the glass vial story and the foreign particles. And I jump to another topic, which is the formulations with micropellets. And these micropellets are a very common dosage form, typically applied in hard capsules, like here, less often as tablets. And here there's a nice insight where to cut off into the hard capsule, and you can see the pellet, the micropellet ensemble. And you can scan this object as an ensemble, or you can do a high resolution scan as a single object. And I like to provide now a case study with the whole ensemble to get some statistical data. But first, I need to explain how the micropellet works. And therefore, we use a layer model and look at the structure of the micropellet, which consists always of a core, typically sugar, and then on top of this core, the API is coated. And then there comes the isolation layer next. This layer is actually protecting the API from the control layer because sometimes they don't go along well with each other. And then finally, the control layer, which is the most important function layer here. It typically consists of some polymeta-acrylates, and those compounds have the beauty that you can steer the solubility depending on the pH value. And this is the nice thing about the micropellets, because in the stomach and in the intestine, we have different pH levels everywhere. So by steering this polymet acrylate nicely, we can actually steer where the drug is being released. So typical products are enteric-coated tablets or sustained release dosage tablets of this. And together with the manufacturer, we work on a defect model for the isolation layer. So this should be our defect. If there is a defect in the isolation layer during the control layer coating, the control layer can touch the API and actually make it inactive. So in real scanning, the defect could look like this. So here's defective isolation layer, and here another one. And in the segmented image, you see these are just open balls. So a quantitative analysis is quite straightforward. By just counting how many of these balls are open, I've presented you the whole ensemble with the defect model visualized in 3D. You can see the micropellets. Now in grayscale, we are coloring the core with the API in blue, and then the isolation layer in green, coming up in green, yes. And the orange layer is then the control layer. And now let's make the core transparent, so we can see whether the isolation layer is actually intact. And we are picking this example. So follow this micropellet. You see, there is some of the control layer sticking to the hole. So this would be one of those defects that I've talked about. And now it just comes down to counting the balls which show minimum 1, 2, or 3. It's up to you. Defects, and then you have a quantitative result of the ensemble, and you can divide them up into defective pellets and intact pellets. In this case, it's quite a significant number. So if we count this, it's 45%, almost 45% defective and 55% intact. That's a pretty nice example of an image analysis and a defect model. And by this, I will close with the solid formulations, because I brought to you one other example from packaging systems. This is only one example because I think time is short today. Packaging systems are used to protect the drugs during storage and delivery to the end user. And I like to make a case study with a sachet. This is a side-sealed pouch bag, actually. And typically the powder inside has to be protected with an airtight sealing because it's a hygroscopic medical powder. And let's have a look at the sealing here. And this is a cross-section of this seam. Yeah? And the question to be answered in microcity is whether there is an open end-to-end channel, which will then provide possibility for air and humidity to cross the ceiling and enter the pouch bag. So XRM can help you to assess this. I've brought to you two examples with a good or intact ceiling and a defective or bad ceiling. So let's start the second one. So in green, I will display here the prep pores, and they are not harmful at all because they are not connected to each other. And then the pores which are connected to a larger network, I will code in red here. And there are still some which microcity tends to mark up as defective, but actually that's still a good theme. But you can see the difference when you look at defective sealing. There is a large chunk being connected from outside to inside. So this is why this sealing scheme has failed, and microcity can precisely find even the location where the end-to-end channel rise up. So this was my last example. I think time is almost running against 30 minutes. I have thank you for your attention, and I hand over to John.
All right. So very, very interesting examples, Marcus. Thank you for giving those.
Yes, you're welcome. Thank you.
All right. And so now what we're going to do is we're going to go ahead and transition. If anybody has any questions, by the way, go ahead, enter those in. We're already getting a lot of them. That's great. So we're going to now transition over, though, from kind of the macroscopic world that we're looking at, or somewhat microscopic, down to the actual kind of atomic scale, or that, well, sometimes we say microstructural, right, Nate?
Yeah.
But that more atomic scale with Nate.
Okay, let's see if I can do this one. All right, I think my slide should be showing. I'm working with a single monitor today, so. Okay. Change my mouse. All right, so thanks everyone. My name's Nate Henderson. I am jointly appointed as the Business Development Manager for Pharmaceuticals for X-ray diffraction here in the US, as well as a Senior Application Scientist specializing in powder diffraction. So similar layout and structure to what Marcus had. A little bit of information about the technique itself for those of you who might not be familiar with it. Some information about instrument geometries, lots of different ways to get data depending on the size and shape of the material that you're looking at, and then a couple of selected examples that we feel are pertinent within this particular industry. So X-ray powder diffraction, so powder here distinguishing it from its partner technique, a single crystal diffraction, which we won't cover today, but of course, very, very relevant within the pharmaceutical space. XRPD is looking at powdered materials or polycrystalline materials. And what we're looking at is we're functionally measuring the distance between atoms or molecules in a three-dimensional space. So what we see here on the right-hand side is a goniometer. So this is one of our D8 advanced instruments. X-ray tube here on the left-hand side, X-ray detector on the right-hand side. Sample goes right here in the middle. And so with this piece of equipment, what we're doing is we are very precisely controlling the angle of incidence between the tube and the outgoing beam diffracting off your sample towards the detector. So very precise control over your theta here, your theta angle here, which is what we see shown in the actual data. So 2 theta along the bottom axis. So you can see here, we're starting at a very, very low angle. So 1 degree, so half a degree on the tube side, half a degree on the detector side. And when we look at diffraction data, we don't quite have the same nice, pretty images that we would see from, say, an X-ray microscopy thing. We simply just get this this line here, and every time we see a peak, that means that there is some characteristic fundamental distance within a crystalline material. So we use things like these peak locations, the relative intensities between the peaks, and then information like the width of the peaks. To massage out some information about what is a material or what is the average crystallite size. Some brief information about the types of geometries that you could see. Most common is going to be called the Bragg-Brentano geometry. This has a diverging beam of X-rays, so X-ray tube here again on the left-hand side. The size of the beam is controlled with some set of slits, either a motorized or fixed opening, and this is going to determine how wide of a beam you have when it hits your sample. This will then diffract off to your detector, and then you will measure the data on the backside. So very, very common. The nice thing about Bragg-Brentano is that you have a very large sampling area. So if you were approaching something like, say, quantification, or you want to look for a very small amount of impurity, the best way to do this would be to increase the statistics of your powders by illuminating more of the material. So you can see there's a little bit of a powder right there in the center of the instrument. So that would be in the center of the sample holder. Another geometry that we frequently see within the pharmaceutical industry is a Debye share or a capillary. So you can see right here, it's a little bit hard to see, but there's a small glass capillary that is filled with powder, which you see schematically here. So a very skinny beam will be created. You'll illuminate your capillary. Often this capillary is rotated to increase your sampling statistics, to change the orientation of those little crystals. And then again, this will diffract off towards your detector on the backside. So much smaller sampling volume here, but capillary diffraction can be very useful if you have material that orients very heavily, or say you have something that is, say, moisture sensitive or air sensitive, or maybe it's a very potent compound that you need to protect by sealing it in a little glass straw. Final geometry I want to mention is a microdiffraction. So this would be a point beam. So instead of thinking about the X-ray as sort of like a light bulb or a flashlight, in this case, we're going to use some sort of collimation or beam shaping to make a very, very small spot. So typically this would be, say, like a 1 millimeter spot or lower in size. And then from this, you get a burst of X-rays and a cone. So we'll see a little cone showing up. And then you would typically couple this with, say, an area detector, like we see here. So that shiny surface here, that whole area is your active detector area. So we'll be able to get large sections of these rings. And this will give us some information about whatever localized environment that we're analyzing. So we see this very highly in the high throughput space. So these would be, say, a 96 well plate, and each one of these little spots would be a different formulation or a different combination of freebase and solvent, or you could do, say, like salt screens. So a lot of different areas that you need to measure using this very, very small spot beam. Okay, so in context of different areas within the pharmaceutical space where we see diffraction really playing a strong role, starting at the very beginning with discovery of new drugs, so high throughput screening for new active ingredients or drug substances, things like polymorphism, looking at different polymorphs, which are different structurals. We'll talk about this in a few slides, different salts, different solvates. Moving from a candidate drug substance over into a drug product, we're looking at things like formulations. So different types of additives, things that can add bulk to a tablet, things that can, say, lubricate for dye presses, colorants or dyes. So if you want to make something a little bit brighter, or if you want to put a shell on the outside of it to make it more recognizable, to increase things like patient acceptance. These can be characterized, again, with diffraction, all the way through to the back end once you have a finalized product. Looking at things like quality control and making sure that you're producing the same thing over and over again. Positive material identification or anti-counterfeiting can also be used. And then looking at things like stability and aging, which could either go into scale up or maybe over here during the development side of things. Looking at your material as a function of temperature or humidity, and really seeing how these materials start to age as a process of sort of external factors. From a diffraction standpoint, these could be sort of segmented out in a slightly different nomenclature. So phase ID would be, what is this material that I'm analyzing with a diffraction experiment? Which polymorph is it? Is it a hydrate or an anhydrous formation? Looking at quantification. So if you have a formulated product, how much of this is active ingredient? How much of it is your each individual excipient? looking at things like crystalline versus amorphous. We can look at things that are either non-crystalline or glassy, or things that are crystalline and give you those nice sharp peaks. Again, non-ambient diffraction for things like aging. Structure determination, this would be for a new material, understanding the actual crystal structure itself, what's the size and shape of the unit cell, and where are the molecules within it. This can also be helpful for things like patenting if you want to really define what type of material or form you've created. And then moving on to things like cluster analysis and large data set processing. So things like high throughput screening, you often end up with a very large number of data sets, and cluster analysis techniques can be really helpful to sort of parse through this and say, which of these are interesting candidates and which of these are maybe something that I've seen before. So we'll go through some of these examples quickly in our time left over. So qualitative analysis, here we have 3 over-the-counter pain relievers. So this is acetaminophen or paracetamol, depending on where you are in the world, aspirin and ibuprofen. So without knowing anything else about them, I can see that they are different diffraction patterns collected from a diffraction experiment. So I know that they're different materials. So sometimes that can be enough of an experiment to say, is this the same or are these different? Things that could also be different are things like polymorphs. So we're here looking at 2 different forms of paracetamol, same molecule, right? But in the case of a diffraction experiment, we're sensitive to not just the molecule itself, but how this molecule arranges in 3-dimensional space. So here we see sort of this wave packing structure. Here we see more of a sheety or a platy arrangement of the molecules. So the polymorph on the bottom that we see here is actually a little bit better at things like direct compression. So if you're looking at tableting or formulating this into an oral dosage, this would be advantageous to have something that is more amenable to direct compression. In the case of acetaminophen, this is often the more common polymorph that we see, this waveform. So this might mean you need to go to something like, say, a wet granulation process or some sort of tumbling. So the polymorph itself can affect things like formulating a product. Of course, it can also affect things like bioavailability and uptake. Most people who are in the pharmaceutical space are very familiar with the ritonavir case, where polymorphism actually very negatively affected the bioavailability of this particular drug substance. So useful information that can be extracted out from a diffraction experiment. Polymorphs, salts, and solvates. So looking at, say, the free base or a salt or a counter ion, or looking at different solvates, whether this is water or any other sort of solvent. This is where we often do a lot of our screening approaches. Here, what we see in the data is theophylline. So the top pattern is an anhydrous form. The bottom one is a hydrated form. So a single hydrate, so monohydrate. So one equivalent of water for each equivalent of the molecule itself. So obviously same molecule, right? But here we do see different diffraction data, which means that there are distinct structures here depending on whether you have the anhydrous form or the monohydrate. Okay, this is something that we could track as a function of time. So this is a wet sample of theophylline, so 100% RH, very, very hydrated material. And what we see is that we complete these scans very quickly. So these were about a 2-minute data collection for each scan. As we continue to scan the same sample, what we see is that hydrated form continues to come in, and the anhydrous form is consumed in that process. So you could tie this with things like vaporsorption, or any sort of supplemental techniques to get a really good picture of what exactly is going in, what is going on in your sample. This could also be done with things like a humidity chamber, where you're directly controlling the RH of the sample. I don't want to spend too much time on these next few slides. This is mostly looking at API or drug discovery. So looking at things like the unit cell. So we know that this is the space group P212121. This is going to be an orthorhombic space group with these lattice parameters, and we can refine out the size and shape of this particular box. So that's what we see here. This is naloxone. So this would be for, say, opioid overdosings. Here we see allantoin. So this would be refining the or structure determination. So if I have really high quality data, I can feed the diffraction software a molecule, and it will flip the molecule around and will try to resolve out exactly where the molecule is in three-dimensional space. So here we see sort of these nice repeating motifs of the allantoin molecule itself in the three-dimensional crystal structure. So mostly looking at things like a single phase, a single material, maybe a new active ingredient. But since the title of this is dosage form and drug delivery, we're looking at things generally on further along in the pharmaceutical process, things that are formulated and may have more than one material. So here what we see is just a sunscreen. I figure it is summer, so it would be useful to have, remember to use your sunscreen when you go outside. But commonly used white pigments that we see in formulations, so zinc oxide and titania. In this case, we see the titania here in blue, the zinc oxide here in red. And if you look very closely, you can see that the zinc oxide peaks are a little bit sharper, whereas the titania peaks are a little bit broader. And so the breadth of these peaks can give us some useful information about the average crystallite size. So here we would expect to see that the titania having a broader peak is going to have a much smaller crystallite, where the zinc oxide having a sharper peak is going to have a larger crystallite. So this could be useful information for formulations. This can affect things like flowability, and then how well a dye may adhere to, say, the exterior part of a tablet. But also, we can look at things. So this is a sunscreen, so it's a liquid, it's a cream. So think about other alternative types of materials that you could analyze, not just solid dry powders, but maybe say a tablet or a gel or a paste or a cream, something like that. As long as there is suspended solid material, particular material, diffraction can give you some useful information. So titania, of course, is a very interesting thing. Very, very nice white pigment. Obviously, there's some challenges in terms of compliance of whether you use titania, where you are in the world in terms of formulation. There are some interesting new excipients coming out that are using calcium carbonate to try to replicate the white color of titania. But here what we see is that with titania, two different polymorphs, this tiny little peak down here is about a 1% impurity of the anatase form. So rutile generally will have a little bit better masking properties, depending on where in the light spectrum you're looking at. So a tiny little bit of this impurity phase can be detected with diffraction. So this can help you back control which type of pigment you're looking at. Looking at, say, tablet engineering. So this is an over-the-counter ibuprofen tablet. What we see is that the inside of the material here is going to be the blue pattern. So that is going to be just ibuprofen. Whereas if we look at the entire tablet that's been crushed up, we see peaks from sucrose as well as the ibuprofen. So we see that the outside part here, if you're trying to reverse engineer this formulation, would be just a sugar shell. And that could be used to provide some calories for settling your stomach. It could be used to keep this this rather fluffy material together in shipping. It could, a structural behavior, or it could be used to flavor a formulation to keep it from tasting too bitter. Other types of excipients, they aren't necessarily crystalline like that sucrose, or here like we see gypsum. So this is a calcium sulfate dihydrate. So nice sharp peaks. Other excipients that we see might be things like glidants or bulking agents, or this sodium starch glycolate. This is a disintegrant. We see a little bit of diffraction from the remaining halite. So this would be sodium chloride. But different types of characteristic broader features here for amorphous formulations. And if we look at this one, so this is data that I collected last week. This is a 1% indomethacin and a 0.5% indomethacin in a predominantly amorphous background. So this is polyvinyl pyrrolidone, or PVP. We're thinking about this in terms of looking at spray dry dispersions and how do you track to see what types of crystalline material you can see forming as a material ages or as it's exposed to different hydration levels. So we see, if we zoom in on this region here, so down below we see the raw pattern for indomethacin. We can see very clearly the 1% indomethacin or the 1% API, and a little bit of features here for the half away percent. So this should give you a sense of scale as to what types of levels of detection you can get with diffraction. So 1% pretty reliably done. Below this, generally on a case-by-case basis, depending on how well you know your material. And then looking at things like, how do I quantify this? So this would be a whole pattern fitting. So each of the colored filled in regions on the bottom are going to be reference patterns that were collected of these individual materials. Blue trace up top is going to be the formulated product. And so we can see that we can scale the exact contribution for each of these materials, even if you only have sort of these broader features to work with. Looking at non-ambient diffraction, so as a function of either temperature or say humidity. In this case, we're looking at lactose as a function of temperature. So this is 2 theta in intensity. And as we go up, we see an increase in temperature here. So starting at room temperature, so 25 C all the way up to 250 C. So this is 2 different visualizations of the same data. So the more traditional diffraction data here on the right-hand side, we see an intensity plot where the brighter the region is, the more intense a peak is. So we see one region down here below, say 130 degrees. Between 130 and 160, we see dehydration. The monoclinic form is here. Conversion of monoclinic to triclinic. Here we see the existence of both forms. And then above 200 C, we start to see a loss of crystallinity or a melt event happening. So very useful information that you can use in conjunction with things like DSC or TGA to really get a full picture of what this material behavior is at a non-ambient conditions here. And then final example would be once you have something like this, where you have a large number of data sets, or in this case, we're looking at several different families, you could apply similarity. So this would be, if you're familiar with things like the principal component analysis technique, this simply applies similarity across your scanning range. And you can cluster your data sets into different regions. So here we see a nice tight cluster for 3 of diffraction patterns with 1 small outlier. And so this allows us to quickly identify one diffraction pattern, which we see, of course, is that extra peak right here at 20. So we could say, oh, maybe this is an interesting polymorph that we haven't fully stabilized. I should go back and look at this particular sample again. Or maybe this particular material isn't pure, so let's throw this one out and focus on the ones that are consistent with each other. So final comments here. So these are examples of the diffraction portfolio, so from small to large. Depending on the type of environment, how many samples you want to put in. But I do want to very briefly show this D6 phaser. This is a fairly new product for us. This is exciting. We just announced it last week globally to the community at large. So you can see it here on the bench top. So this will be the successor to the D2 phaser, which we've seen a lot within the pharmaceutical space, but much higher power, new capabilities for things like capillary diffraction, non-ambient diffraction, looking at things in transmission, dynamic beam optimization with things like a motorized optics. So we're really excited about seeing this and what people in the industry are going to be able to do with it. For more information, of course, you can go to bruker.com/D6 phaser, or feel free to reach out to us after the webinar and be more than happy to put you in contact with some great application notes or some more information. So with that, I will turn it back over for a Q&A session.
All right, thank you, Nate. We'll bring everybody back up here. And we definitely have a lot of questions that have come in, so we're going to try to get to as many of those as possible. So the first question that we have that has come up is, porosity analysis, is it qualitative or quantitative measurements by XRM?
Yeah, both actually. It's using the segmentation method, we can get precise volumes and size distributions. But some customers just look for large pores, which are like singular defects. That's more qualitative.
All right. And then the next question, what is the minimum size of samples, pores, which can be analyzed by XRM? Would it be useful for 100 to 500 micron single component nanoporous granules?
So depending on the instrument that you have, the best possible spatial resolution is in the low 0.1, 0.2 micron range. So nanoporous, you have to define how nano this is, but it can be tricky sometimes. If it's below 100 nanometer for sure, that is away from X-ray diffraction limit.
All right. Next question. Is it possible to measure the coating thickness of the pellets? I think you may have talked about that a little bit.
Yes, that is also very possible, just in the same way image analysis I've shown for the coating of the tablets. Yes, we can do it.
All right. Question kind of for me, will the slides be made available? What we'll do is we're either going to send out the slides or we'll send out a recording of the event.
Yes.
So watch for an email on that. Lyo cakes, is the analysis, is it destructive or analyzed from outside of that vial?
Yeah, 10 years ago or 8 years ago, it used to be destructive. So you would stamp out from the cake and put it to the microscopy. But now with the new technique available, we scan it non-destructively within the glass. So that's fine.
All right. We're gonna do this. We're gonna jump a little bit here to some of the XRD questions. Let's see here. What's the best and most sensitive XRD technique to detect the amount of crystalline API in a product containing primarily an amorphous?
Tricky. The best technique, honestly, is a good old Bragg-Montano. If you were looking at a small amount of crystalline API, the challenge is often homogenization. You want to make sure that the material is fully illuminated by the beam. And the best way to make sure that you're analyzing everything is to hit it with a very, very broad beam. So one of the things that we're seeing more often is the use of a variable divergence slit rather than a fixed divergence slit, which actually increases the size of the beam as a function of scanning angle. So it keeps the whole sample illuminated, which can really, really help out in terms of overcoming some of these challenging statistics.
All right. Thank you for that one. And then another XRD question. What's the least LOD that can be achieved and the factors to be considered to achieve the most stringent limits of detection?
So the answer to every good scientific question, of course, it depends. We have gone down as low as 1/4 weight percent. That did require us to have a immaculately prepared sample and one that diffracted quite strongly. So we did, so a nice sharp peak. With the indomethacin example, 1/2 weight percent, I think, is detectable if you know what you're looking for. Which requires you, of course, to also look at just your amorphous material or, say, your excipient blend. These were obviously very simple cases, so one excipient, one API. But if you had, say, like a very engineered formulation, you would need to be concerned about things like peak overlap. And so the requirement to have a clean peak off to the side is going to be part of determining what LOD you can reach. But usually we say 1% by weight can be done. And from there, you kind of work your way down based on how well you can push it.
All right. Now I've got a question actually for both of you. And that one is, what is the X-ray power and time of exposure used for XRM? I know that X-ray exposure can yield to degradation of pharmaceuticals when doing XRD. And I think this kind of depends a lot, like what's in the question here about the power and exposure times, right? So maybe let's start with the XRM side of things.
Yeah, so typically the lab sources that are used in XRM tabletop instruments for tablets are operated around 50 kV, and the maximum power, primary power on the target is 10 watts. So and then probably efficiency, the target is 50%, and another 50% is gone on the way. So probably we are ending up to send 2 watts of X-ray flux to the sample. very rarely I've seen degradation. It could happen for long scans, but normally it's not so big problem in XRM because we are anyway looking only at the structure. I mean the microstructure, not the structure of the molecule which degrades. Typically will not change at all. But for example, with amorphous lenses, we will see they get some browning after some time, which is really a molecule change, but not so, anyway, not to be seen in X-ray micro-CT.
Okay, so Nate, what about the XRD side of things?
Yeah, so I think that often people, so say like a beamline, a synchrotron, like a very, very intense X-ray beam, like certainly you could see some browning and some damage to the sample. I think in a laboratory setting, we often don't see this. We don't see it frequently. And for anyone who's done anything within the radiation side of things, that it's time and power. It's the length of exposure time. And so I think it's one of the things that is really cool about modern equipment is that data tends to be a bit faster because of advances in things like sources and technologies and things like that motorized diversion slit. You can get through your data much faster before something negative might happen, like X-ray damage. But at least in a laboratory setting, I don't frequently see this on samples that I encounter.
Okay, yeah, and we don't have a SCD or single crystal diffraction expert necessarily here. I know that they use maybe a little bit more focused beams, right, where you might get a little bit brighter, tinier, more flux in a certain spot, so you might have a chance.
And I think that the statistics part of that also comes into play, right? Because with a single crystal experiment, you've got one, right?
Yeah.
And you hope that nothing happens to it. Whereas in powder, often we can kind of take advantage of the fact that there's so much material there.
Yep. So if anybody out there has that question about SCD in particular, go ahead and reach out to Bruker, and we can get you connected with an expert in that field also. All right, next question, also kind of coupled. How do you couple the XRM and XRD results? The grayscales are related to density in XRM. How can we find the crystalline structure that the gray color is related to?
Okay, I'll take a stab at it. All right. So typically, I find that it's not fully forensic. It's not fully diagnostic, right? Like I would never take a material and just say like, I don't know anything about it, right? Like with microscopy, you can get things like maybe the more dense particles happen to be your API. Maybe there's a metal center there, or maybe it's just it's more granular or more compacted, so that you have the sort of the excipients that are going to be fluffier, and you would know that. Then I would take, say, the same tablet, I would crush it up and throw it in a diffraction experiment, and I would see a signal from everything that is in there. So I would say, maybe I've got a broad feature from microcrystalline cellulose, and I have some small little sharp peaks. I could then go back in and then try to identify those sharp peaks and say, which API is it? You could try doing things like selective dissolving and recrystallization, but I think that it's often the case that we aren't fully trying to solve an unknown. Particularly with formulated products, it's not I have no idea. I mean, there's so much paperwork, right? There are safety data sheets, there are drug disclosures. You often know what is in there. You're just trying to figure out exactly, can I confirm it, rather than what is this peak? Yeah.
Marcus, any thoughts?
Yes, yeah, of course. It's important to understand that XRM is just looking at density differences, so basically absorption differences. So there's no real sensitivity to the molecule unless you use some more modern technique like phase contrast, which I have skipped for today. But there can be tablets that consist only of, from atomic standpoint, only of carbon. So there are no contrast at all, except, for example, cracks, pores, and inclusions or something. So it is not a magic tool, but it is certainly a very valuable and powerful addition to your lab. So a direct comparison between XRD and XRM is not as straightforward as you would think.
So, and I guess there might be some inspiration too from the world of geology. In that case, what people are looking at is two methods. One is looking at pure components. So you'd look at, say, in this case, the API, you'd look at the excipients, and then you would take that pure phase, throw it into the XRM, and you may be able to get some idea of that density contrast. What does it look like relative? The other bit is, if you do have the mixture, you could do certainly the quantitative analysis. You could say segment the whiter areas, segment the grayer areas, the darker areas, and then compare that quantitative analysis to your XRD quantitative result, and you may find some very strong correlations. So we do, yeah. So with that, we had a lot of questions come in, and what we'll do is we'll put them together actually into a questions document, and then send that out along with, like I said, either a recording or a copy of the slides. So thank you for sending all those in, and thank you everybody out there for attending. Of course, thank you to Nate and Marcus. That's very insightful things here.
Thanks, John. It was a pleasure.
Okay, and if anybody out there does have more questions, anything further, please feel free to reach out to Bruker or RJL, and we can help you out with any of your questions. So with that, go on, and if you're in part of the world, you can have a wonderful evening. In another part of the world, you may have a wonderful day today, and we will see you at our next event. Goodbye.
Thanks.